Boa tarde a todos. Se você quiser, eu vou dar minha palestra com o sotaque português de Portugal. Talvez não. Lembra, ontem eu falei que é necessário para vocês falar in inglês e entender inglês. So I'm going to switch to English because I think it's important for me to remind myself of my mother tongue and to speak in English. So let's see if the presentation is ready. Not quite. Minutinho. So that was a good session, that last session, I thought. We do those type of journal clubs back home. And in fact, I didn't know some of the slides I'm going to show were also in that journal club. This sort of ambitious title, we have some really distinguished people in the audience. One of my heroes is Mike Lederman here, and he has educated us about the immune response to HIV for a long period of time. And so he's actually got a great little video for his journal, Pathogens and Immunity. I would suggest you Google and have a look at. You'll hear from Mike later on in the course. But is Mike's sort of inspiration that made me come up with this title, Can the Immune System be Trained to Eliminate HIV? But then I thought maybe a better title is Immune Targeting of the Latent Reservoir. And you've already heard quite a bit about cure. You're going to hear a lot more about cure research. There is only one person that we know cured of HIV, and that's Timothy Brown. And that's an extremely interesting story. And of course, a lot of investigation is going towards trying to replicate that. You'll hear from Dan Kuritskas of his exciting work. And you've already heard from Matteo and Rafik. And we really want to try and cure this. So just a refresher, you've already heard a bit about HIV latency from the pre-course, from talks today. There are a number of potential ways in which we think we can wake HIV up from its latency. Histone deoset lasers, David Margolis made this slide, so these are some of his favorite drugs. But there are also other ways that can work at different stages of this pathway in order to wake HIV up from a resting CD4 cell. So Lucio just showed you this slide. It's the same slide, I just made it bigger and didn't put the full reference. But there is this long decay in people who are treated with antiretrovirals for their latent reservoir in CD4 positive cells. And so this means that you'd have to keep someone on antiretroviral therapy for a very, very long time to try and eliminate all of these last residual cells that harbored virus. And so a number of different strategies have been provided and discussed, and you'll hear a very exciting one from Susanna Valenti. But in most cases, we think that we have to get viral production back in order to kill all these residual cells. So we have to wake all the latent cells up and then have different methods of killing them. Lucia also talked about broadly neutralizing antibodies, and BNAbs are probably a critical component, as well as cytotoxic T cells and other ways of the immune system to try and make the immune system work better against the viruses that are woken up after latency reversal. So kick and kill, shock and kill, you've already heard this phrase, you'll probably hear it a lot during this course. You have the integrated HIV, you have a latency reversal agent which wakes that HIV up into production, that's called a shock. And then you have a transcriptionally active cell, and that transcriptionally active cell can put peptides into newly formed MHC molecules to be flagged on the cell surface by cytotoxic T cells, and also new viral particles presenting GP120 can form on the cell surface. And then you can kill these with various immune or other drug effects, and the idea is that this will help in the elimination of the latent reservoir. So a few years ago, working with Mario Ostrowski at the University of Toronto and Brad Jones, who was a grad student of his at the time, we thought about what might be impediments to this immunological recognition. And we know that HIV is highly variable in its sequence. And so we wanted to try and think of a way which we could target an infected cell that was looking at very conserved epitopes, or potentially a unique antigenic target that might be expressed in that latent cell once it had been woken up. So just to remind you about the way that cytotoxic CD8 cells will recognize an infected cell, you have to have newly formed processed peptides that nestle in the groove of a newly formed MHC molecule, and then that complex is taken through the Golgi to the cell surface, where that specific peptide from a viral antigen in this case stands like a proud Brazilian flag on the surface, ready to be recognized by the Supreme Court. So our question was, is there a stable, fixed antigen that could mark an HIV-infected cell and target that? In other words, could we find a unique marker of a cell that had been latent that would allow us to target these cells for killing? So I'm going to tell two stories today. One is about human endogenous retroviruses as unique antigens expressed in latently infected cells. And then I'm going to talk about some other antigenic cell molecules which others have identified as potential unique markers that could also potentially be targeted. So when we go back to our biology class, it's remarkable that we are taught mostly about a very small, slim pizza slice. Imagine if you went to a pizzeria and they gave you that slice, right? You'd be pretty hungry. And all of the rest of this is other parts of non-coding RNA, long interspersed nuclear elements, signs which are associated with lines, and then these endogenous retroviruses, which make up about 9% of everyone's genome in this room. So between 5 and 9%, it's interesting that even though we have the whole genome project, that people can't actually yet settle on what percentage of the genome is endogenous retroviruses, but it's reported to be between 5 and 9%. And the transposable element family, for which endogenous retroviruses belong, has a number of different members. You have transposons. They're quite rare. They transpose through DNA intermediates. You have transposon through RNA intermediates, but don't encode a reverse transcriptase. You get the retropseudogenes, like the allo family, and then you have those that do encode reverse transcriptase, and you have those that lack long-terminal repeats, the retrotransposons such as lines, and then you have those that have long-terminal repeats and many elements of retroviruses, and these are the endogenous retroviruses, or the retrovirus-like elements, including the ERVs and the one family. So endogenous retroviruses in our genome, 5 to 9% of our genomes. You have a long-terminal repeat. You'll have as a minimum this sort of reverse transcriptase, but you can also have other gene products. So you may think, why does our genome have these fossil retroviruses sitting in it? And we think that they are the fossil remnants of past infectious retroviruses like HIV, past infectious retroviruses that became integrated into the chromosomes of germ cells and are passed down from mother to daughter or child. And we have these present throughout most of our genome. There are interesting areas of a couple of chromosomes which seem to lack endogenous retroviruses. But we think that these are evidence of past infectious retroviral infections. And if we think about it, humans have been impacted 3 times in what is called a holobiontic event in human history. So more recently, humans were colonized, if you like, with mitochondria. The microbiota, a topic of great interest, and endogenous retroviruses are thought to have come in about 150 million years ago into ancient lineage. So these sort of invasions of organisms make us wonder what a human is. You know, are we really trillions of bacteria plus these fossil viruses with a few extra fillings? So endogenous retroviruses can be in a number of different phylogenetic groups. Here we have HIV, and there actually are some endogenous retroviruses that we have recently discovered that are quite similar. So we're in the process of working that out, and we'll publish that hopefully soon. But you have endogenous retroviruses such as HERV-W that are part of the gamma retrovirus family, and then you have HERV-K, thought to be the most recent of the insertions in the beta retrovirus family. And when we think about, this is millions of years, you know, this is a long time ago, 100 million years ago, as the primates split, oops, I'm not sure why we're getting the feedback. I'll try and stay very still. It might be the, that's right, it's my squeaky jeans. Thank you, Lucio. Oh, I have to walk. Okay. I have to, let me get my water. Maybe I'll stand back here. So, each time there is a split of the apes or the primate families, there are new insertions. And actually, we had a little speculative paper that in hominoid evolution, each time the hominoid evolution split, there's a new insertion of an endogenous retrovirus. So, of course, without any evidence at all, one can say that there is, you get driving speciation from endogenous retrovirus insertion. And so, you know, what's the next one going to be inserted? We don't know. Woo. Here we go. No, it's different. Okay. Squeaky jeans. So one of these things about endogenous retroviruses is there's really no sensible taxonomy system. And I think there needs to be one. And I'm going to talk a little bit about the way we're approaching this. So they've named after the host tRNA involved in reverse transcription, HERV-W, HERV-K with a lysine, HERV-L, leucine, etc. But sometimes someone, it's a bit like naming a star, right? You get arbitrary names, and HERV-FRD. I mean, you know, it's like Fred. That's the Fred HERV. Now, HERVs are interesting for a number of different ways, and one of the discoveries that really was quite interesting about 15 years ago now was that the envelope protein of HERV-W actually encodes a protein called syncytin, which is expressed in the placenta and is involved in the formation of the syncytial trophoblast. So humans have co-opted endogenous retroviruses in some cases to do good things, and human pregnancy wouldn't occur unless we had this HERV-W protein. So, as I briefly mentioned, you get infectious retroviral epidemic, endogenization, you gradually get increase in HERV copies, and then eventually these become fixed in the population. in addition to HERV-W and the placenta, there are other examples of domestication of HERV sequences. They actually turn out to be incredibly important in human embryonic development and pluripotency. This has recently been discovered by a Stanford group, and they're working actively on that. But also, endogenous retroviruses have been postulated to be important in a number of different infections or different diseases. So I said that most of the chromosome has these. There's this interesting, the Y chromosome is pretty bereft of these, and there's area of chromosome 1, which doesn't have many HERVs, but they're scattered around throughout the genome. Let me just go back. So if we just think of one of the HERV families, HERV-H, there are almost 5,000 of these scattered out. So when we talk about HERV-H or HERV-W or HERV-K, they're not all one HERV we're thinking about. They're scattered around. So there's no reason to think that these aren't behaving in an independent manner. And in fact, we now know that they are really part of the active RNA regulation in cells. You can get enhancement, they can act as alternative promoters, they can have a number of different impacts, and some of them are actually long non-coding RNAs. So some of these endogenous retroviruses are long non-coding RNAs. So why study HERVs in HIV infection? Well, what we're interested in is how could we beat antigenic variation, because that makes the design of something that's going to hit a latent cell that's been activated difficult. And if we think about the scale of HIV variation, this very nice slide which shows this is the variation within influenza virus. Here's within one HIV-infected single individual. Here's a cohort. But when you look at the Congo, with all its amazing different clades, you can see that there's a huge scale of HIV variation that needs to be taken into account. So to get back to the question that I asked, is there a stable, fixed, non-HIV antigenic target that could mark an HIV-infected cell? And so our model was with Mary Ostrowski and Brad Jones that because HIV has this protein, you see I talked about VIF, which takes APOBEC to the immunoproteasome for destruction. APOBEC wasn't there to deal with HIV. APOBEC was probably there to deal with some of these endogenous retro elements within the genome. And so by releasing APOBEC, by taking APOBEC away, we hypothesized that you would get endogenous retrovirus reactivation. So it's like, poking around in a fossil pit and then finding some magic agent that would allow new endogenous retrovirus antigens to be expressed. And the advantage that we thought is that this could then mark an HIV-infected cell, and only an HIV-infected cell, and they would be fixed, non-mutatable sequences. So Brad Jones and Keith Garrison in my lab, when I was in San Francisco, did a number of studies where we showed that HIV-induced HERV-K transcription, and also could see this in vivo. Brad, with Mary Ostrowski, went on in collaboration with us to also show that primary CD4 cells infected with different HIV isolates could express endogenous retroviruses. And then even more remarkably, we found that we could use a HERV-KN specific clone to recognize these endogenous retroviruses. It was independent of VIF and could recognize a number of different clades. So the basic model seemed to be correct. We could have a system where these endogenous retrovirus-specific immune cells could recognize HIV-infected targets independent of the clade. But we were concentrating on HERV-K, but you'll already, from my HERV-H diagram, be reminded that there are many different HERVs. So when I say HERV-K, we might think, well, there's one coding sequence. There isn't. There are 90 different HERV-Ks scattered throughout the human genome. And we didn't know which ones might be affected. So how do we resolve HERV expression for specific genomic locations? And we took advantage of RNA-seq technology. So this was done by qPCR. People looked at endogenous retroviruses with microarrays. But these ERVs, if we go down through the class, the family, the subfamily, the genomic copy, to individual elements, we needed a better tool in order to assess which endogenous retroviruses were being differentially regulated. So Matthew Bendel, a graduate student in my lab, who's a bioinformatician working with colleagues in Mexico City, used RNA-seq to develop a computer pipeline to address this problem. So with RNA-seq and looking at transcriptomes, it's usually easy to make alignments of those fragments to your reference transcriptome. But because transposable elements are present in many copies throughout the genomes, it provides a challenge in order to identify which fragment belongs to which particular endogenous retrovirus. And so we have these cases where there's unique hits and those that are ambiguous. So bioinformatically, you can develop a program called the EM, the Markov model, where you iterate your data and basically allow you to calculate based on the probability which will go with which particular fragment. And at the end of this program we call Telescope, we can uniquely map endogenous retrovirus fragments to where they came from on the genome. And so we now have a tool that we can look at any RNA-seq data and say these endogenous retroviruses are upregulated, downregulated, or remain the same. So the advantage of this RNA-seq data is you can go to the literature and you can look at RNA-seq libraries. And we looked at primary CD4 cells from a Bushman paper with Cheryl Mix, and then two cell lines. And the first thing, when we looked at what we're calling the retrotranscriptomic index, which is the count per million of map fragments of the retrotranscriptome, if we look at primary CD4 cells, we were gratified to see that indeed in an HIV-infected CD4 cell, there's more endogenous retrovirus expression. And interestingly, in the transformed cell lines, there's also a lot more retrotranscriptomic activity, and that might be expected in the context of a transformed cell line, dysregulated gene expression. You might expect there to be more endogenous retrovirus expression. And we could look at the number of expressed loci and identify ones that were identifiable. So here, for example, if we look at the cell lines, we see that there are certain endogenous retroviruses which are differentially expressed. They're either upregulated or downregulated. And in primary CD4 cells, we see several that are upregulated. Note that what you see in a primary CD4 cell is not the same as what you see in a cell line. Another reason we should study primary cells. And we were actually very gratified because independently, Ben Mulder at Mount Sinai School of Medicine found that the same endogenous retroviruses were upregulated as we had found from the RNA-seq data, sort of independent validation. So the findings from this is that cellular models of HIV infection are different from primary models. We've identified some ERV loci that are activated during HIV infection, and with this program we have a novel approach for looking at differential expression of these transposable elements. And we've been looking in HIV-infected latent cells, and hopefully I'll have some data on those soon. So while we were working on this approach, others have been working on different ways of identifying latently infected cells. We know that latent cells predominantly are in CD4 positive T cells. There's probably an important monocyte macrophage compartment which is understudied but is real and needs to be looked at. But a lot of data from the CD4 positive cell subset shows that there are enrichments of a number of different markers. But how do you get a unique marker? So we were, like many of us, excited by this paper published in Nature from Monsef Benkraouen and Benjamin Decor. I'm not pronouncing their French names as well as I could do. But they published this high-profile paper suggesting that CD32A is a marker which actually can identify these HIV reservoir harboring replication competent proviruses. This was an extremely important paper because if we have a marker that can identify those latent cells, we can think about pulling them out. We can think about killing them. So people were really, I think, very excited about this. So we, and I say we, Richard Apps in my lab, in collaboration with James Whitney at Beth Israel in Boston, looking at humans or looking at macaques, asked the question, can we replicate that data? Can we use CD32A as a marker of latently infected cells? So straightforward, can we detect 32A expression? Does it correlate with reservoir size? Is this a useful marker? So from the DeKelp paper, they found in the CD32A high population, this is where the bulk of the HIV DNA copies resided. So this was a really important result. And they showed that CD4 T cells depleted of CD32 expressing cells had negligible amounts of replication-competent virus. And so on the basis of those two results, they concluded that CD32A identifies over 50% of the reservoir containing CD4 cells. This is fantastic. This is what people want to see. However, in our hands and in other people's hands, this is not what we observed. So in humans or in rhesus macaques, in PBMC or lymph nodes, we sorted CD32 high or negative cells, and we didn't see any significant difference in the proviral DNA content. So that goes against what they had discovered. When you look at the percent of CD32A high cells in relation to proviral DNA, there wasn't any correlation, again, between this population and proviral DNA in PBMCs, lymph nodes, or even gut. In rhesus macaques, CD32 is expressed prior to SIV infection. The frequency doesn't change immediately following infection. And that contrasts with the in vitro system, which Ben Crane used, which showed no prior expression to infection. Of course, this is rhesus macaque. It's different from humans. There may be differences there. But in tissues as well, there's no consistent relationship with CD32 cells and a reservoir marker. So we didn't see that. And again, one thing that we wonder and might have misled Ben Crane's group is that these CD32 high CD4 cells are highly activated and express a lot of DR. And that's also inconsistent with what we mostly think, in that quiescent cells harbor latent reservoir. And then James Whitney and his group looked at the expression of CD14 and found that in the system that they used, there was a lot of B-cell contamination in the CD32 high population. And so that could potentially explain that result. And then there were also these CD14 monocytes, which they hadn't excluded. And so part of the reason that we think they might have found the result is that they hadn't potentially excluded all the contaminating cells that they might have done. And then in humans, we wanted to look at the infectious unit of the reservoir using the QVOA assay. And the amount of replication-competent HIV wasn't associated with the frequency of CD32 CD4 positive T cells, and this also was true in lymph nodes. So in our hands, the expression profile of CD32 is inconsistent with being a reservoir marker. We found that CD32 high cells, CD4 positive cells, don't contain a significantly higher proportion of proviral DNA in either the SIV or HIV systems. There's no correlation with the replication-competent HIV. And we think that there may be error within the flow cytometric measurements. Now, of course, this is a pity, but and it's also, showing you negative data, because sometimes negative data is as important as positive data. We have to, if someone's result is going to be robust, it has to be replicated by a lot of other people in the field. And the majority of studies which are not yet published but are coming out suggest that they have similar findings to us, that CD32 does not mark the reservoir, which is a shame because, of course, we're all looking for a unique marker. So I'd like to stop there. A lot of people to thank. Brad Jones, instrumental in a lot of this work. Matthew Bendel, my grad student who does the bioinformatics. Richard Apps, Miguel, Nate, John, Greta Yanking. And then a large list of collaborators, Mario Ostrovsky from the University of Toronto, my colleagues in Mexico City, and James Whitney at Beth Israel. So thanks very much indeed.
And right now we will just open for questions because Doug will need to leave us right now. We have like 5 minutes to discuss. Anna.
Thanks, Doug. I'm always intrigued with your targeting ERVs as a means of identifying cells infected by HIV, and it's a lovely concept. But in terms of adapting or in harnessing host defenses, how much selfness is there to an anti-ERV response?
So that was one of the reasons we wanted to better identify which ERVs are expressed, because in a cancer cell, there are a number of different ERVs expressed, but we did some extensive pathology studies which suggested that there was minimal reactivity against self cells. Now, that could always, is an important, if not the most important consideration to think about targeting ERVs. ERV therapy has been used. There are antibodies against ERVs. There's an antibody against ERV which is used in treatment of multiple sclerosis, and there's no bystander impact of that. And then Richard Childs at the National Cancer Institute has targeted ERV-E in renal cell cancer, and there hasn't been any negative impact on that. And then the Wang-Jo Hannings are targeting ERV expression in prostate cancer, again, early days, but doesn't appear to be any deleterious effect. But one of the reasons we wanted to better understand which ERVs are expressed is to more effectively target. And then there may be other ways we can target ERVs outside of the standard immune approaches. We could use aptamers, we could target RNA expression. So I think our approach has been, we've seen this phenomenon, now let's discover exactly which ones are being expressed.
So, Doug, do you think those endogenous retroviruses play a role in negative selection, like in mouse? And are they conserved throughout evolution, or humans have very distinct human endogenous retroviruses than mouse or non-primates?
So it's a little different to the mouse. We know that different mice congenic strains have different complements of endogenous retroviruses, to start with. Secondly, the mouse has an exogenous life component, so you can get an endogenous retrovirus is being reactivated, forming an exogenous retrovirus, which can then reintegrate and endogenize. So the mouse is a little different. Endogenous retroviruses are highly expressed in the human thymus, and I don't think anyone has really worked out what role they may play, but they're very highly expressed in human thymus in the fetus, and so there's probably something going on there. We just haven't investigated it ourselves. But the embryonic stem cell people are onto that, because from the Nature paper, there's this wave of endogenous retroviruses that come up and down as each cell divides, and there probably is quite a lot of things going on there that we still have to discover.
Although there is a precedent with NZB, NZW model of murine lupus, where actually RT inhibitors are curative. So it turns out that maybe in some autoimmunity there is actually an effect of a retroviral antigen involved. So it's not tolerance, complete tolerance then. And actually it's interesting because people have been trying to do GWAS studies with lupus, have not really been able to get anything but MHC class 2, and some people believe this is because it may be related to endogenous retroviruses, which are not the same in the population, so you cannot actually assign a SNP very easily by this kind of genetic mapping.
Yeah, so I've been working on that with colleagues in London, and we merged our telescope program with MAGMA, which looks at GWAS SNPs, and so we created a program we call Magnoscope, and we've looked at SLE and have identified around 25 HERVs that are differentially expressed that we're investigating.
Anyone else? No. Well, thank you, Doug. Thank you very much.
Thank you.